In this video, we will explore the impact of defects in RNA splicing in various diseases. We will discuss mutations in splicing factors in myelodysplastic syndromes, dysregulation of non-coding RNA in neurodevelopmental disorders, and splicing defects in mitochondrial diseases. We will also cover treatments such as antisense oligonucleotides for splicing defects, mis-splicing in cardiac channelopathies, and splicing defects in intellectual disability syndromes. Myelodysplastic syndromes are blood disorders that can progress to leukemia. Mutations in splicing factor genes can disrupt normal gene expression, leading to impaired blood cell production. Targeted therapies are being investigated to restore normal splicing patterns and improve patient outcomes. Research has shown that non-coding RNAs, including microRNAs and long non-coding RNAs, are dysregulated in neurodevelopmental disorders. This dysregulation can lead to splicing defects, disrupting the normal expression of genes involved in neurodevelopment and contributing to the manifestation of these disorders. Understanding this relationship is essential for developing targeted treatments. In summary, RNA splicing defects are emerging as important contributors to mitochondrial diseases. Mutations in splicing factors or regulatory elements disrupt mitochondrial transcript splicing, leading to abnormal protein production and impaired mitochondrial function. Further understanding of these mechanisms is crucial for developing targeted therapies. Antisense oligonucleotide-based therapies have emerged as a promising strategy for correcting splicing defects in genetic diseases. These therapies use synthetic oligonucleotides to target specific pre-mRNA sequences and modulate splicing outcomes. By binding to the pre-mRNA, antisense oligonucleotides can change splice site recognition and correct splicing defects. Successful applications have been demonstrated in diseases like Duchenne muscular dystrophy and spinal muscular atrophy. Challenges such as delivery to target tissues and off-target effects remain, but ongoing research is advancing the development of more effective oligonucleotide-based therapies. Cardiac channelopathies result from abnormalities in ion channel function in the heart, which can cause arrhythmias or sudden cardiac death. Recent studies show that mis-splicing events in genes encoding ion channels contribute to these disorders. These events lead to the production of abnormal channel isoforms, disrupting electrical properties and increasing the risk of arrhythmias. Understanding this mechanism is essential for developing targeted therapies. Intellectual disability syndromes are characterized by cognitive limitations. Splicing defects in genes important for neurodevelopment and synaptic function contribute to these syndromes. Mutations in splicing factors can lead to abnormal splicing and gene expression, resulting in cognitive impairments. Studying these mechanisms is important for developing targeted therapies. Duchenne muscular dystrophy, DMD, is a severe muscle disorder caused by mutations in the dystrophin gene. Splice-switching therapies have shown promise by targeting specific gene regions to restore production of functional dystrophin protein. Clinical trials have demonstrated their potential, and the FDA has approved the first therapy for a subgroup of DMD patients. Challenges in delivery and mutation types need to be addressed for wider application. The process of alternative splicing allows for the production of multiple mRNA isoforms from a single gene. RNA binding proteins, RBPs, regulate this process by binding to specific RNA sequences and structures. Abnormal RBP expression or function can lead to diseases, including cancer and neurological disorders. Understanding how RBPs influence alternative splicing is important for developing treatments to correct splicing defects. Muscular dystrophies are genetic disorders affecting skeletal muscle, causing weakness and wasting. Splicing defects play a crucial role, producing abnormal mRNA isoforms that affect muscle-specific proteins. For example, in DM1, MBNL proteins are sequestered, leading to splicing misregulation and disease features. Understanding these mechanisms is vital for therapeutic development. Hematological malignancies like leukemia and lymphoma are caused by genetic changes in the splicing process of RNA molecules. Mutations in the spliceosome complex disrupt normal gene expression and can lead to the development and progression of these diseases. Understanding these mutations is important for developing targeted treatments. Cancer splicing patterns are influenced by RNA binding proteins, RBPs, which interact with pre-mRNAs to modulate splicing outcomes. Dysregulated RBPs can lead to altered expression of cancer-associated isoforms, affecting tumor progression and treatment response. Understanding these regulatory networks can inform targeted therapies. As we age, changes occur in our bodies that can lead to a decline in physical function and increased risk of age-related diseases. Recent research has revealed that alternative splicing, a process that creates different versions of genes, plays a role in these processes. These splicing patterns change with age and have significant effects on cellular functions. In diseases like Alzheimer's, abnormal splicing can contribute to the development of key disease features. Understanding the molecular mechanisms behind these splicing changes is crucial for understanding aging and age-related diseases. Identifying splicing regulators and specific events associated with aging could potentially help restore normal splicing and prevent age-related diseases. By delving into the complexities of RNA splicing defects, we uncover crucial insights into the molecular underpinnings of diseases. This understanding paves the way for targeted treatments that rectify these defects and enhance patient well-being. Ongoing research concentrates on alternative splicing regulation, spliceosome mutations, and the involvement of RNA binding proteins in disease mechanisms.